Cellular respiration is how we drive energy from the food we eat, especially from glucose. In order to turn this glucose into energy, we require the 6 oxygen molecules. Through cellular respiration, we turn glucose and oxygen into 6 CO2 and 6 molecules of water and ATP. This is the formula of aerobic respiration, which is glucose and oxygen will become carbohydrate, add water and energy. Anaerobic respiration proceeds the absence of oxygen and does not result in the production of any further ATP molecules. When ATP becomes ADP, the energy released because of the bond between phosphate group, a high energy bond. When one phosphate leaves, the energy is released. And since there are a lot of water molecules just floating around nearby OH pairing, that's called a hydroxide, from one of the H2O comes over and take place of that third phosphate group. It's called hydrolysis. The whole process of cellular respiration is take place of cytosol to mitochondria. Mitochondria are the power plants of the cell, synthesizing large amount of ATP via aerobic respiration. The structure of mitochondria are basically intermembrane space, outer membrane space, inner membrane space, and matrix. Glucose is transformed into ATP over 5 separate stages: glycolysis, link reaction, the Krebs cycle, electron transport chain, and last one is chemiosmosis. These stages are described as coming one after the other. From cytosol to mitochondria, through cellular respiration, 1 oxygen molecules and glucose are yield 38 molecules of ATP.
Glycolysis is the breaking down of glucose 6-carbon ring into 2 3-carbon molecules called pyruvate molecules. This process can be summarized by 4 key points. First one, called phosphorylation. A hexyl sugar is phosphorylated by 2 molecules of ATP, which makes the molecules less stable and more reactive, and also prevents diffusion out of the cell. Next one, called lysis. The hexyl biphosphate is split into 2 triose phosphates. Next one, called oxidation. Hydrogen atoms are removed from each of the 3 carbon sugars to reduce NAD+ to NADH plus H+. Finally, called ATP formation. Some of the energy released from the sugar intermediates is used to directly synthesize ATP. This direct synthesis of ATP is called substrate-level phosphorylation. In total, 4 molecules of ATP are generated during glycolysis by substrate-level phosphorylation. But in glycolysis, glucose needs 2 ATP to split into 2 3-carbon sugar. The net gain of glycolysis are 2 ATP to ANDH plus H+, and to pyruvate. And the next stage called link reaction is also the first stage of aerobic respiration, which transports pyruvate into mitochondria. So, pyruvate is transported from the cytosol into mitochondrial matrix by carrier protein on the mitochondrial membrane. The pyruvate loses carbon atoms which form a carbon dioxide molecules. The 2-carbon compound then forms a acetate group when it loses hydrogen atom by oxidation. NAD+ is reduced to NADH plus H+. The acetyl compound then combines with coenzyme A to form acetyl-coenzyme A. For per glucose molecules, the link reaction produces 2 acetyl-CoA, 2 NADH plus H+, and 2 CO2.
The next step of aerobic respiration is Krebs cycle. Krebs cycle is the one of important step of aerobic respiration, the step after the link reaction. During this process, the Krebs cycle will do 2 times because there are 2 pyruvate inside the glucose. And this cycle will produce some FADH and NADH and ATP. The FADH and NADH will be used in ETC process. First, the production of link reaction, acetyl coenzyme A, will come into this cycle and bind with an oxaloacetate, which is a 4-carbon component. This kind of binding will cause the carbon of acetyl-CoA come off and bind with the 4-carbon component, able to form a 6-carbon component, which called citrate. After this kind of binding, the empty acetyl-CoA will back to link reaction and bind with carbon again. Second, during this step, the NAD plus will come to citrate and become to NADH by the reduction reaction, and the citrate will lose 1 carbon because of the oxidative decarboxylation. In this case, the carbon will combine to oxygen molecule and form a carbon dioxide. After those reactions, the 5-carbon molecule will keep going on to the next step. Third, this step is just the same to the step 2, so the 5-carbon component lose 1 carbon, able to form a carbon dioxide and NADH, and finally become the 4-carbon component. Fourth, this step is called rearrangement. During this step, the ADP will come with another P, able to form the ATP by the substrate level phosphorylation. Also, one NADH and one FADH is formed by the reduction reaction. NADH and FADH will carry the electron and hydrogen for the ETC process, just like a key. Finally, at the end of this cycle, the 4-carbon molecule which is oxaloacetate, will back to the beginning and do one more loop, because one glucose have 2 pyruvate.
The final step for the cell respiration is ETC, which is the electron transport chain. First is the generating a proton motive force. The hydrogen carriers such as NADH and FADH are oxidized and release high-energy electrons and protons. The electrons are transferred to the electron transport chain, which consists of several transmembrane carrier proteins. As the electrons pass through the chain, they lose energy, which is used by the chain to pump protons, H cations, from the matrix. The accumulation of H cation within the intermembrane space creates an electrochemical gradient. The next step is ATP synthesis by osmosis. The proton motive force will cause H cations to move down their electrochemical gradient and diffuse back into matrix. This diffusion of protons is called chemiosmosis and is facilitated by the transmembrane enzyme ATP synthesis. As the H cation moves through ATP synthesis, they trigger the molecule rotation of the enzyme synthesis in ATP. The third step is the reduction of oxygen. In order for the electron transport chain to continue functioning. The de-energized electrons must be removed. Oxygen acts as the final electron acceptor, removing the de-energized electrons to prevent the chain from becoming blocked. Oxygen also bonds with free protons in the matrix to form water. Removing matrix protons maintains the hydrogen gradient. In the absence of oxygen, hydrogen carriers cannot transfer energized electrons to the chain and the ATP function is halted. That's all of cell respiration.